Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today's video is related to RNA sequencing data analysis, and the title of my today's video is How to Identify Differentially Expressed Genes from RNA Sequencing Data. So as you all know that I have started a video series related to RNA sequencing data analysis, and this is the last part of that entire video series. So in today's video, steps and concepts you are going to learn in this video that what is heat map and how to interpret a heat map. Then I will proceed with that with what is PCA plot and how to interpret a PCA plot. Then I will also show you that how to build PCA plot and heat map from gene expression data using R programming. After that, what is DEGs and why we use it, and how to find differentially expressed gene from gene expression matrix file. And after that, in the last visualization, we will visualize the differentially expressed gene through different plots like Volcano plot. And I will also teach you that how to interpret a Volcano plot. So basically today's video is a little bit descriptive because I want to cover all the concepts, and these all concepts are linked to each other. So that's why I'm explaining and performing these concepts in a single video. So this video will be a little bit descriptive. If you know some of the concepts related to these concepts, so you can skip some parts of this video. But I recommend that we have traveled a long journey. We start from the very beginning of RNA sequencing data, and now this is the last part. So it is very much important for you people. Secondly, it is also important to start the video series from the very beginning if you are new to this channel or this playlist, because I have explained each and every step in more detail related to RNA sequencing data analysis. So I start from the very beginning that what is basically RNA sequencing. I discuss the NGS data and the NGS experiment, and how to read what is FASTQ files, the Linux operating system, and each and everything. So this is a complete playlist of 18 videos. Okay. So I tried my best to teach you RNA sequencing data analysis. So I assure you that if you start from the very beginning, your time will be not wasted, and you will learn a lot of things related to bioinformatics, especially RNA sequencing data analysis. So in the previous video, I teach you that how to arrange, upload, and transform gene matrix file in RStudio. So basically, today's video is linked to that video. Okay, so I recommend to watch this part 17 and then part 18 of this video, because the data I have already uploaded the data to RStudio, and I will be proceeding after the previous code. Okay, so that's why I recommend you to watch this part, then skip to this part. Okay, so let's start with the explanation and the theoretical and the practical stuff. So first of all, as I have mentioned, I will teach you that what are heat maps and PCA plot, and then I will show you that how we can build heat maps and PCA plots using gene expression data. Okay. So principal component analysis or PCA plot is basically a very complex concept, and it involves a lot of steps, statistical steps that are used to build a PCA plot. But in a very much simple way, PCA plot is nothing but just it show the variability of your data and compare two phenotypes related to the variability inside their data. Let me explain. For example, you are having two samples, cancer and normal, and in cancer, these both phenotypes are related to gene expression data. Okay, so cancer sample also have gene expression data, and normal samples also have gene expression data. Basically, I am talking about this data. Okay, so for example, you are having three samples of normal individual that are having specific gene expression data of these genes, and similarly cancer phenotypes also have gene expression data related to these genes. Okay, so now you are having the gene expression data of both the phenotypes. What basically PCA plot do is that it calculate the variance inside the data and filter out the highly variable data from the highly variable gene expression data from the gene matrix file, and after that it compares both the phenotypes with respect to the variability inside the data. Okay, so basically it's comparing gene expression data and showing that how much these two phenotypes are distant from each other with respect to high variable data. Okay, so basically if you are having cancer and normal phenotypes, so these dots are basically the gene expression data. Okay, so for example, if there is a specific gap inside between two points, so we can say that according to the variability of the data, the gene expression data, these two phenotypes are distant from each other. For example, this blue point is present here and these red points are present here. So we can say that there are some gene expression values between the cancer and the normal that are very much distant from each other. So basically PCA plot give you cluster related results and explain partially that how much your gene expression profiles between the two phenotypes are distant from each other, different from each other. So it's very much simple. It gives you a clue of your data, okay, that how much it is distant from each other. So this PCA1 is basically principal component 1, and this is principal component 2. So on the x-axis it is PCA1, and in the on the y-axis it is PCA2, and on the x-axis it is PCA1. And 40% of the variance is explained on PCA1, PC1, and 14% of variance inside your data, gene expression data between the cancer and the normal, is explained on y-axis. So about 14 plus 40, so 40, we can say that 54% of the variance of your data is explained by this plot. Okay, so you can get a point from it. So basically, this is PCA plot. I will show you that how to build a PCA plot from a gene expression matrix file. Now, what cracking the concepts of heat maps? Now, there is also concept of heat map, and heat map is nothing but it basically it shows the distance between two phenotypes, okay, or two samples. So, for example, why we construct heat map is that, for example, we are having sample 1 normal and sample 2 is from cancer, and sample 1 also consists of gene ABC, and same is the case with sample 2. The same genes are also present in cancer sample or sample 2. Now gene A in normal have a specific gene expression, and similarly gene A in cancer sample also have a specific gene expression. Okay, the same is the case with gene B and gene C. So how much these samples are distant from each other considering gene expression values? Okay, so we are checking the distance, the difference between two samples with respect to each gene expression. Okay, so very much simple. In PCA plot, we were choosing those gene expressions that were highly variable inside the data, or we can say they were highly different between the samples. But here in heat map, we are considering each and every gene data. Okay, we are not looking to the highly variable gene expression data. We are completely looking to each and every expression of each gene. Okay, so that's why PCA is a little bit different from heat map, and heat map can give you a high level of information related to PCA plot because it is considering each and every gene expression. Now, the PCA plot, heat map is basically built on different statistical formulas, but there is a specific distance formula that is known as Manhattan distance. And mostly when we are constructing heat map, so when we are calculating distance between two samples with respect to their gene expression values, so we use Manhattan distance formula. So basically, this is the formula of Manhattan distance. Now, I will show you that how gene expression are placed inside the Manhattan distance formula and how the distance is calculated. Okay, so very much simple. Now here we know that this is the Manhattan distance formula, and y and what are y and x. So I will show you. So for example, we are having sample 1, 2, 3, 4, and there are gene A, B, C, D inside each sample. Now the value of gene A inside sample 1 is 10 inside, and the value of gene A inside sample 2 is 12, and same is the case with sample 3 and 4. Very much simple. Also gene B, the expression value is 15, in sample 2 it is 18, 14, and 16. Now, how we can calculate distance between sample 1 and sample 2? So very much simple. Now let's calculate the Manhattan distance between each pair of samples. So the distance between sample 1 and sample 2 is that 10 is basically belongs to this the gene A, the value of gene A in sample 1, and 12 is the value of gene A in sample 2. So 10 minus 12 according to the formula y1 minus x1. Very much simple. The same is the case with 15 minus 18, 20 minus 22, and 25 minus 28. So basically these are the genes value, these are the genes, and these are its value, and we are subtracting the gene expression values from each other. But we are also taking the absolute because distance can never be in negative. So in order to cancel the negative effect of the distance, we take absolute of it. So now the distance between sample 1 and sample 2 is 10. Same is the case with sample 1 and 3. It is 8, 5, 12, 7, and 7. So we calculate all the possible distances between all possible samples. So now, after that, when we get these results, so then we have to place it inside a heat map. So very much simple. The distance between sample 1 and sample 1 between a same sample is always 0. We all know that, for example, 10 minus 10 is 0, 15 minus 15 is 0, 20 minus 20 is 0, 25 minus 25 is 0. So the overall, there is no such distance between sample 1 and sample 1 because they are similar. Now, the distance between sample 1 and sample 2 is 10, and we calculate it here like this. The same is the case, the distance between sample 1 and sample 3 is 8, and the distance between sample 1 and sample 4 is 5. So all the similar concept will be followed for all the samples, and after that, the value of the distance will be placed inside a heat map. So for example, if we and we can then also color it to different colors in order to visualize it effectively and differentiate between two samples. So very much simple. 12 is the highest distance, and we can say that 5 is the lowest distance. So red color is basically related to lower distance, and green color is basically related to higher distance. So it depends upon you that what color palette you are choosing to show the distance between two samples. So this was all about heat map, and now this is a type of heat map that we can construct from our gene expression data. So, but the concept behind this heat map is exactly the same as I have discussed. So the color basically shows the value of the distance. So basically it is just like this. So if you want to see these numbers inside the color, you can do it. And if you want to just see this colors inside the blocks, so you can also do it. And you can place a color palette or a reference that one can relate it, that if it is lighter color, so there is higher difference, and if it is if it is darker, so there will be no such distance difference between it. So for example, as I have told you, sample 1 and sample 1 have zero distance. So basically the diagonal is completely dark color, because dark color means zero. So very much simple. It is not that much difficult enough. You all have to just go through this concept, and it can be easy for you. Okay, so this was all about the PCA plot and the heat map. Now I will show you that how to construct PCA plot and heat map using RStudio. Okay, so basically we in the last video we left from here. We upload our data, and we just transform our data using log transformation. And these were the gene expression values, as I have shown you in the picture, that how it looks like. So basically these are the genes. Basically these are the genes, and these are the samples. So these are the samples, and these are the genes, and these are its values. So very much simple, as I have told you, to construct a heat map, you have to calculate distance between each and every sample using the gene expression data of each and every gene. So very much simple, calculate sample to sample distances. So sample distances is an object, and distance, first you have to transpose your assay data. Assay data is nothing, just this data. This data is known as assay data, because it consists of these genes and these gene expression values related to a sample. So first we will transpose it. Transpose means that we will place these samples in rows and these genes in columns. Very much simple. You do not have to do it manually. You just have to do it using RStudio. So we will transform it, and after that we will just calculate the distance between it. So very much quickly, they will calculate the distance between, if you are having 50,000 genes, as we are having 50,006 genes, so it will calculate the distance between samples. We are having 10 samples. So considering 10 samples and considering the 50,006 genes, it will calculate the distance between using the Manhattan distance formula. And after that, we want to visualize it. So this is the complete code of it. All you have to make changes according to your data if you are having a data. So all the code will be the same. You just have to replace this part. Okay. So in the metadata, if your phenotype column is, if it is basically another heading, so you will place the another heading basically here. So now I want to visualize it. So first of all, you have to arrange the data. Now sample matrix, row names, column names. We set the row names, the column names. We tell that the row name should be of the phenotype, and the column name, we do not need the column name. We do not need the gene names. And after that, we will apply a palette, and after that we will construct our heat map. So very much simple. Now we have constructed our heat map. So basically these are the cancer normal. Basically this is the phenotype, and this is the palette, and now we can interpret it. Okay, so as I have shown you the interpretation. After that, we want to calculate the PCA plot. So very much simple. Plot PCA is a function that consists of all the steps that are related to calculating the PCA plot. Okay, so basically these are the steps as I have shown you: get data, compute mean, this covariance, eigenvalues, eigenvector. It all calculates it in a single function, and after that it is calculated. And very much simple, as I have shown you, using the 500 top feature by variance, because I told you that variance should be used. And now this is the plot. Okay, let me show you, as I have also shown you before, but now. Okay, so basically this is the plot. So group cancer normal, and this is the PCA plot. So very much simple. You can construct your PCA plot according to your data. You all have to just change this phenotype if in metadata there is the heading is changed. Now after that, a very simple concept, perform DESeq analysis. Now this is the very important analysis, and this DESeq is used to find differentially expressed genes from your gene matrix file. Okay, so first of all, before finding differentially expressed gene, all you have to do is some little bit calculations, not calculation, but some consideration. You all have to do is to convert your phenotype data, these cancer and normal data, into factors. So in order, so it would be easy for the DESeq object to read it. Okay, DESeq tool to read it, or package. We can say DESeq package. So very much simple. We convert the phenotype data into factors, so it could be easily read by the DESeq package. After the re-level to make normal appear first in the data. So this is very much important. You want to compare your gene expression data related to normal. So basically this is the reference, and we all need that. For example, if you are comparing normal and cancer, so it's up to you that you have to compare what should be your reference. So it depends upon you. For example, if you are performing an experiment or a calculation, and you are having that we can say a different reference. If you are using a different phenotype, for example, Crohn disease and ulcerative colitis, for example. So Crohn disease, you want to make Crohn as a reference or ulcerative colitis as a reference. So very much simple. It depends upon you. So we want to compare the gene expression value of cancer related to normal. So this is our reference. So what we will do is basically we will relabel it and we will consider normal as our reference. So now we have considered normal as our reference, and if we want to see is basically we are having 5 samples of normal and 5 samples of cancer. Now all we have to do is that we have to convert the normal value to factors in order to be easily rated by the DEC package. So this should be a little bit done. Okay, so it is basically performed to ease our analysis. Now the main step is run DEC2. So it will take a little bit time. We will write DDS and we will apply DEC on our gene matrix file. So very much simple, the analysis is started. So estimating size factors, estimating dispersion, gene-wise dispersion estimates, now these are calculating. Let me tell you some important stuff about DEC package. Basically, this is used, it is a complex statistical package that is used to find differentially expressed genes inside your gene matrix file. So very much simple, if we look here to this matrix, so we do not know that which genes are highly expressed and which genes are lowerly expressed in our sample. But according to statistics, if this DEC is designed to find that differentially expressed genes. So very much simple, it will do all the thing for you. You all have to just wait for it and apply the correct code. So basically it uses moderated estimation of fold chain and dispersion for RNA-seq data with DEC2. You can use this paper, study this paper, and you can, if you want to know about the statistics behind it, so you can visit this paper, and it is clearly mentioned that what type of algorithms they have applied to make this package and to be usable by the user. Okay, so if you want to know about the deep statistics, so you are welcome. You can just go to the Google Scholar and search about it. So I'm just showing you the practical. And if you are still concerned about that, how does it work? Just let me know in the comment box. I will make a separate video related to DEC2 to explain the statistics of DEC2. Okay. Now after that, now it is performed. It performed gene-wise dispersion estimates, mean dispersion relationship, final dispersion estimate, and after that they fit the model, and then they found the differentially expressed gene. Now let me tell you what the table look like. So inside the table is basically, now we will just we perform DEC analysis. Now we will make the final table from it. So very much simple, res make an object and apply this command on this DDS tool. So results DDS. After that, it will take a little bit time because after huge statistical analysis, it will make a specific table. So it will take a little bit time. And after that, when the table is generated, now we will observe it. So now the table is generated, and now observe important information from the table. So if we want to see the row names, so the column name, so basically it consists of base means, log 2 fold chain, LFC, stat, p-value, adjusted p-value. If we want to summarize it, so basically according to the summary, it consists of 11%, 0.034% of upregulated genes and 0.043% of downregulated genes. And it also shows that there are 1.5% outliers and low counts are 7.5. So this is the entire summary of our gene count, and out of it, it consists of 32,000 genes. Okay. Okay. After that, we apply some filtration. We want to see the genes that have statistical significance of p-value lower than 0.05. We want to see the genes that have expression values we want to see the differentially expressed genes having statistically significance. So we will apply this code, and after that we will see the summary of it. So basically after applying this code, we will see that there are no outliers. So we were just removing the outliers. Now we can also see the head of the table. So basically this is the head of the table. These are the gene names, and these are the base mean, log FC value, LFC, error value, stat value, p value, and adjusted p value. I will try to explain each and everything. We can also see the descending order. Okay, so now this is the ascending order. The highest value is 8.63 of HLA-DOB2 gene. Okay, so basically 8.623 means the log FC value 6.67. 8.67 means that it is highly expressed in cancer samples. Okay, because we consider normal as a reference, so the positive value will be basically for the cancer samples. Okay, now after that, if we want to see the top gene, this gene, if we want to plot this gene, so very much simple, just plot it, and after that, now we can plot it here. So let's see, let me explain. In normal, the expression is according to this bubble plot, this is the expression. But in cancer, its expression is very much high. It reaches to 500. Okay, so this is why the log FC value of this gene is higher, 8. So it is higher in cancer sample. Now very much simple. Let's proceed with the final calculation. We want to see all the differentially expressed gene, the significant and the non-significant differentially expressed gene. So we will construct a Volcano plot for it, and this is the entire code of Volcano plot. All you have to do is just change these subtitles and titles according to your sample. You are not required to change nothing at all. You all have to just change these parameters. If you want to apply a cutoff of more than this, so you can change this. You can also change this if you want. You can change this, these, but the entire script on the syntax is the same. After that, you can also change this title and subtitle and caption and these colors also. It all depends upon you, but the script is the same. Now I want to apply it, so very much simple. It will use this entire table, the differentially expressed gene table, and it will construct a Volcano plot. Okay, so I will explain that what is basically a Volcano plot, because it also take a little bit time to construct. Okay, so it will take time. After that, I will show you that what is a Volcano plot. But before this, we will also make the last table of it in order to show you in the Excel sheet. Okay, we already construct the Volcano plot. I will explain it in a minute. After that, apply these codes to make your final table. And now write.csv, we want to see the final table that consists of the differentially expressed gene. So we will write that in a CSV format inside our directory. Okay, so it is written inside our directory, and basically I will show you. Now, but first of all, I want to explain that how to interpret a Volcano plot. So very much simple. This is normal versus cancer differential expression analysis. And okay. Okay, so this is normal versus cancer differential expression analysis. This is basically a Volcano plot. Okay, this is Volcano plot. Now, how to interpret it? Basically, it consists of all our 50,000 genes, but that 50,006 genes, but we want to see that what are statistically significant differentially expressed genes. So it basically we want to visualize it, and we want to show the significant differentially expressed genes. So in order to check the significance, we are here for the Volcano plot. So these are our parameters, okay, of the significance. Now, what are these genes? The first significance is the log 2 fold chain. Okay, as I have mentioned, the log 2 fold chain is basically, if it is higher, so it means that this gene is higher in cancer sample, and if it is in negative, so this means that a specific gene is lower in cancer sample. Let me explain. For example, we are having gene A, and its log FC value is for example 8.64, and we are having gene B, okay, and its log FC value is minus 2.64. So what does this mean? If we have made the reference normal, so after that we can say that gene A, the value of gene A is 8.64, and if the normal is the reference, so this means that it is 8 times higher in cancer sample. And if it is minus 2.64, so this means that this gene is 2 times higher in normal sample. So this is basically log FC value. So we apply the cutoff of the log FC value. And for example, if it is minus 1 and plus 1. So we say that we do not need those genes that have log FC value in between minus 1 and plus 1. So basically we remove these genes, okay? So this area is basically, it is not significant according to our log FC value. Now the second parameter is this part, okay? This part. So basically this part and this part. So these are basically related to statistical significance. We want the statistical significance of 0.05. So when the genes that are, oh sorry, this is the 0.05, okay? So the genes that are present below this are not statistically significant. So very much simple, these genes do not follow the two significance. They do not lie in between, they do not follow the log FC value threshold, and also they do not follow the statistically significant p value, okay? So these are completely of no use. After that, these genes, the dark blue genes, basically these genes have statistical significance, but their log FC value is in between minus 1 and plus 1. So we also do not need these genes. Now the yellow genes, although they fulfill the criteria of log FC value, but they are not statistically significant, so we just eliminate these genes. Now these are the red genes. They follow both the criteria. They are statistically significant, and they also follow the log FC value threshold. So these red genes are the best genes that we can consider it as significant differentially expressed gene. So basically this was all about the Volcano plot. Okay, now I will show you the last table of it, and we will conclude our RNA sequencing data analysis after seeing the table. Okay, so this is the table that I have downloaded, and these are its parameter. Basically, this is the gene. Okay, this is basically the genes, and these are the base mean, log FC value, LFC stat, p value, and adjusted p value. So this is the final table. You can go through this table, play around it, apply some thresholds, p-adjusted threshold, p-value threshold, the statistical significance, because this consists of the entire 50,007 genes. Now you are required to apply some statistics and eliminate non-significant genes from this table. Okay, so this was all about the RNA sequencing data analysis. I think if you are following from the very start, so basically you have learned a lot from this, and if you are new to it, so I recommend you to start this tutorial and you will learn a lot from this tutorial, from this playlist. Okay, so thank you very much for today. Inshallah, I will come up with a new tutorial with a new concept related to bioinformatics in the next video of this channel. Inshallah, stay tuned, and if you understand this video, so share it with your friends, like this video, and subscribe to my channel for more interesting videos related to bioinformatics. Thank you very much. God bless you.